Only mode.
Hello everyone, and thank you for attending today's webinar. My name is Brendan Robinson. I'm an area director here at Golden Helix. The title for today's webinar is Identifying Clinically Relevant Variants and Creating Customized Reports. And our presenters are Dr. Ahmed Alfares of the National Guard Hospital in Riyadh, and also one of our field application scientists, Hauwa Yusuf, will be showing some of our VarSeq software as well. So at this point, I'm going to go ahead and turn it over to Ahmed, and he can start his presentation.
Well, first of all, can you hear me? All good?
Yes.
Good, excellent. So I would like to thank you, Brandon, for the introduction. And my talk today, well, again, I work in Saudi Arabia, where there is a high rate of genetic disorders in general, which could be as a result from a high rate of consanguinity, and that's where we apply and use genetic testing more or less frequently as one of our first-tier testing, especially when we came across patients with atypical phenotype. And one of the more frequently ordered tests recently is the whole exome sequencing. So we started this in 2011, whole exome, and then with the time we start by sending out samples outside, and as the time progressed, we get more knowledge and understanding, and more tools became available for better interpretations and analysis of the whole exome. Recently, what we do only outsourcing the wet part of the exome sequencing to a commercial laboratory, and then we receive the raw data files, which is either could be compressed of FASTQ, BAM file, or down to the VCF file. And this is one of the things that what you can see on my screen is filtration, a customized filtration steps that have been implemented into our laboratory for a VCF file analysis. So just to be clear, we don't do sequencing until now. Soon, hopefully in 6 months, we just received the equipment. But we do the analysis and interpretations, and also we have our own pipeline. So that's just a walkthrough about our workflow steps. When we receive a FASTQ file, which is not frequently, sometimes we just go to the VCF, then we need to check a few parameters that we are okay with that, and then we do the alignment phase. We generate the PAM QC file that we want to make sure that we meet with our criteria, that parameters that the PAM file has passed it, and then we do we call the variants, and we use also different three variant caller tools, and then we generate our FASTQ file. Either we use the GATK or SAM tools or FreePass, and that's basically until we come to what we will be focusing more in today's talk is about what we call the filtered, or we name it as a filtered VCF file. So when we reach this, that's where we start loading our samples into Golden Helix. And I will show you now how we created the customized tools and filtered in order to, after loading the VCF file, and where we can pinpoint to a single one variant, and then how also we worked with Hauwa in designing our customized report that will fulfill our requirement. So a variant that when we have this filtered VCF file, a variant that we want to filter out anything that is more than 0.1 in frequency, 0.01 in frequency. There is an additional 0 here, which shouldn't be. And then we go back to this again, our filtered VCF files, and then sometimes we are dealing with that step is basically sometimes we are dealing with the whole genome. So we have some samples with a whole genome sequencing, and we want only the variant encoding regions or in the genes. And then we sometimes expand our filtration process and include intronic, silent, or the variants in UTR. I think which is the most important thing is here where we reach at this stage. So we have this processed VCF file. The first step we do, I'm not sure if the screen is aligned or no, but when we have this processed VCF files, that means we, using Golden Helix or VarSeq, we filtered the allele frequency, we filtered based on the location and the variant of the gene. We came up with this processed VCF file. Our filtration process start. Have we seen this variant in ClinVar before or no? This will be easy, straightforward. If a variant has been seen, just check mark, yes, we have seen this variant, we report it. And then we just make sure this variant is pathogenic. It's really related to the disease or the phenotype of this individual. And if we are satisfied with the classification, there's enough evidence about this variant, this VCF file literally will take you, or this sample literally will take 5 minutes. It will be reported 5 to 10 minutes. And then you're done. Then you can move on to the next sample. If this variant is not in ClinVar, there are many steps. How would you approach your filtration? Either you can go with autosomal recessive dominant, either by mode of inheritance, or the allele stats, heterozygous, homozygous, or the type of variation. So we have been in discussion with our team here, which one we start. Do we go first thing with an allele state, homozygous, heterozygous variants, and then we look into the autosomal recessive, then we look into the missense variants, for example, or we start with the mode of inheritance, and then we go to the allele stats, and then it's really, we think going by mode of inheritance would be the best approach, especially dealing with an area where there is a high incidence of autosomal recessive disorders. From our experience, it's account around 80% from all of our diseases detected by exome sequencing are autosomal recessive. So that would be the best. We think this would be the best approach based on our culture and region. However, this may not be applied to everywhere or somewhere else. And just to make sure that we have not forgot everything, if we also look into the digenic 3' and 5', and I will show you some examples now. So this is how we started our filtrations. Like when we have an autosomal recessive variants, we look for, is this a homozygous or it's a heterozygous or compound heterozygous? And that's exactly what's here. If it is a homozygous variant, we need to look during our filtration process, we look into all these loss of function variants, all of missense variants, the polyphen splicing. We do the same thing for the compound heterozygous. If we are dealing with autosomal dominant, we look for heterozygous and homozygous. And also if we are dealing with the X-linked, we look into male and female. This is just what I'm trying to show you here is our workflow for filtrations. And that's then we have as a quality control that we almost try to cover everything from the FASTQ file until we report this variant. Now, if I will just move my screen to show you one of the cases that I will walk you through two cases, how we used the Golden Helix software to do interpretations of a variant. I'm not sure if the screen is clear now on Golden Helix in the VarSeq software.
Yep, we can see your screen.
Okay, so I already loaded the VCF file and our workflow. I really like this software and it's really making our life to somewhat easier, streamlined. So when we load the VCF file, this is a technician will load the VCF file, and the first thing that the technician will see will be a report that we created, that a template for a report that's called whole exome sequencing, and then the technician will fill out all these data and information about the name, the date of birth, gender, ID, from where the samples, and then the technician usually will stop somewhere here, and then we'll just do saving, and then that's mean this case is ready for interpretation. A geneticist, a molecular geneticist, or a genetic counselor, or someone who is trained to do this process will come and log in and open this file again and to start the filtration process. Because we are using a very customized template that really fits our need, we already, when we load the VCF file, this will be few things that will be clearly filtered out, that nothing genes, read depth is 20, and then we go by frequency. Anything in the database that is more than 0.1% will be filtered out. All of these filtrations will be done just by after loading the VCF file. So if we start here, we are starting with around 60 something, 68 sample, 68 variant, and all of the variants with a high allele frequency will be cleared out. We ended up with a 9,000 variants. However, there are two reasons why someone would expect this number should be around 400, 500, 2,000, 3,000, or 6,000 max. We think there are two reasons. One, these variants it's not based on our database. We are using ESP or EXAC or 1000 genome, which may not be a true representations of our population. Two, we are using 3 different colors, and sometimes a variant called in one color but not the other 2 colors. We don't consider this variants. We only consider variants called in 2 colors. However, it's still counted in the total number of the variants. And now, when the geneticist log in, he will end up starting maybe from these 9,000 variants. And if I just go through the same workflow that I just showed you, this is how we are looking for. The first thing that we grouped the first filtered based on zygosity and ClinVar filters. And this case, I will just skip the ClinVar and I will come back to it later, but I will start with the inheritance, and I will focus, for example, in only autosomal recessive disorders. So I'll be looking only in genes or found in a disease that we consider as an autosomal recessive. And then the next step, based on our algorithm, I'll be looking only in the homozygous variants, and that is 46 variants. And then let's say I want only there are no loss of function variants. However, there are 10 missense variants. So I will choose, I want to see these 10 missense variants. And then these are just, I'm just walking through the first steps of our workflow. Not that I should come back into these just in case if we did not found the variant as faster than what we expect. As you may know, your ultimate goal is to find the disease-causing variants in the less amount of time. So if I may, and that's exactly how we designed our workflow. We are hoping that we will be able to pinpoint to this single variant as much as the faster we can save time. However, if the variant is negative and we still are unable, then we will be looking into all these other details as a continuous evaluation. And now I will just take you back. So we are looking into a variant in the autosomal recessive disease. It's a homozygous. We couldn't find any loss of function, so we'll be looking for only the missense variants. And for a missensitive variants, we look at prediction tools. So we included the polyphen and anything in polyphen that's considered as damaging. So it contains damaging. So that's mean there is only one variant. And let's see if this is the variant that we would consider or no. Usually I will just click on this variant. I will go into details, and this will give me many information and details about this variant. For example, just most of the times I just go directly I look into one important thing is the transcripts. So it's a missense and missense and two transcripts. That's mean it's not a loss of function another transcript, which you would expect to have more severe effect. If this variant has been seen at a frequency, yes, this variant has been seen, and then I just go down straightforward. That's how way this variant is in ClinVar. We did not look in ClinVar, but it's in ClinVar. I usually go to the phenotype, so it's the patient. Sorry, I forgot. The patient is an ataxic. He's a 3 years old with an ataxia. That's very important information I forgot to mention, sorry. So here, if we look, what are the phenotypes? If you look, for example, if you click on the Genome, it will just take you directly to the OMM page. And then if you click on the phenotype, and then you can see that the clinical synopsis, and then it's autosomal dominant recessive, joint contractures, kyphoscoliosis, congenital dislocation of the hip. This does not really fit with our patient. If you look into the OMM, this does not really fit with the patient phenotype. Sorry, the page was like a new page. I was not sure if you can see it or no. But it was like a pop-up window. But that's basically like one variant that it does not really fit with our phenotype, and then that's mean we need to go back to our filtrations and look again. So what we do usually, if we look, that was the autosomal recessive, it's a homozygous variant, it's a missense variant. Is there any other variant that I want to look? Indel, sometimes we do and look for indel variants. But for interest of the time, I will just skip this. It will be detailed step. If I go again to, it's an autosomal recessive disease, and then let's say we'll be looking for a heterozygous variance. The number is higher than what you want to see. Is a 1,300 variant. That's too high for assessment. Sometimes it will be very difficult. And then let's see effects, and then I just click on loss of function variants, and then okay, we have now only 9 variants. That will be more easier to assess these variants. What I am looking here, I am not really looking for the heterozygous variants. I am looking actually more for what I would expect to be a compound heterozygous, and VarSeq would be able to do compound heterozygous assessment if you have a trio samples. However, from our experience, perhaps Hauwa and Brendan can correct me, if you have only a single case, we do it more of a manual curation. So here we'll be looking, that's exactly when I look into this variant, I am looking for a compound heterozygous because we are looking more or less at an autosomal recessive disease, and then it's a loss of function, so prediction has nothing here, has no impact here. And if I go again to our gene list, these are 2 genes. I will be looking at 1,2 gene and 1,2 genes, where someone would expect, yeah, these 2 variants. So we have deletion here, we have a deletion here, and here also we have a deletion, and here we have an insertion. So one would expect, oh yeah, these could be a potential compound heterozygous variants that could affect that could be the reason for the patients to have an ataxia. If I look into first variants, again, this page will be updated with all the details and the information. And this is good. The phenotype is mental retardations. The patient is not really mentally retarded, and that's only mental retardations. Also, we have created a Genome Browser where this is one of the powerful tools also at the VarSeq, where you would sometimes want to visualize what's really happening. We included ClinVar variants, CMVs, if there's a deletion or duplication in this region. We included the frequency, so you can just by visualization for people who love visualization or visuals to have an idea about where are you, or if you are near an exome neutron junctions, if there is a deletion or duplication in ClinVar CMV. It's just an additional tool that we more use it sometimes for visualizations, for additional information, that is if you don't like text. So this variant is not, again, going back to the FMN2 variant, is not really fits with our phenotype. If I go down, 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 I see, oh, this is an SACS variant, another variant. It's a heterozygous. Sometimes I look, just make sure it's frame shifting to it's a loss of function variant. And if I go back, if I go down, 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 oh, this variant and the phenotype. So this variant is Charlie-Lavoie-Saguenay syndrome, where it's basically spastic ataxia with autosomal recessive disease. And we have one variant here, and we have another loss of function variant here, which is an insertion that is an all transcript. It's a loss of function. And if I go down, it has not been seen in any allele frequency. And again, it's also the same disease, the same gene. So we really think this is the real disease-causing variant. And with all the demonstrations, with going through all the steps, it almost took me like 10 to 15 minutes to pinpoint to this variant. However, when the first time we assessed this variance around a month ago, I just started with I started with the total number of the amount of the variant. It's at 9,000 variant, and then I went to ClinVar. I chose pathogenic. I want only to see these 9 variants, and that is our pathogenics in ClinVar. I can see 1, 2, and it took me 5 minutes literally to report these variants, because it was really clear fits. It's reported as one of the pathogenic variants in Charlie-Lavoie-Saguenay ataxia, spastic ataxia, and it's reported clearly as pathogenic. If you want me, Hauwa, to go through how we are using our report, I can do that. But what we do, we create a new variant set. We call this, for example, pathogenic. And then we label it with red. We include this variant, this variant. We go into our report. So it's a positive. We add our interpretation here. We select our variant set, then which variant you want, and then all this information will be pulled out and populated from many sources, and that's then we just generate our report. We are working with Hauwa now to customize this and make it looking better and collect more information, and I believe Hauwa will have some talk about this later also. So this is one thing that we are doing. The other tools that we are using with the VarSeq, let's say you did not really came up immediately using, that was not straightforward case, I think which is a very useful tool, is using the filtration by the phenotype. I'm sorry, just so I can't do this, but what we do is we'll be looking at only the variants, that is where we can score variants by genes. So samples, perhaps I can do this. So this is how, what we also, an additional tools that we have been using is we need to filter based on the phenotype. So let's just go back back into autosomal recessive. I will just clean the ClinVar filter, and we'll be looking only at the autosomal recessive. And so this is and then from these autosomal recessive diseases, sorry, I'll be looking at heterozygous, and then loss of function. So this is the 9 genes if I filtered based on the phenotype. I don't know, sometimes it happens. So sometimes, like, this is what we did. We did here is we sorted by the phenotype. I could show you better in the next case, but well, we'll be looking only for ataxia. And then this is a long list, but we based on, like, this gene is really very associated with ataxia, and then the list goes the number as we go down, how number 1 is the highest number, and as you go down, the chances of the association of this gene with ataxia is decreased. This is my first case. The second case I just wanted to show you. It will be faster, hopefully, because I will not go through all these details. And then I will give the mic for any questions, and also how to show how some report customization will work. So it's just loading. I'm not sure if you can see the screen now or what exactly. But because to move from one case to another case, I don't think you can open 2 cases at the same time using Golden Helix. At least from my experience. So can you see the screen now?
Yes, we can.
Okay, so this is the second case, and in this case it's a microcephaly case. Again, when the technician will load this case, it will open directly into this. It's the same, almost the same thing. He will fill out all this required information. We'll leave it. Then the clinical geneticist or the molecular geneticist will come and do the filtration assessment. And the same workflow, basically, we only included the de novo variant by Mendel errors, where it count a variant that presented only in the index but absent from the father or the mother. If we go the same thing for ClinVar, because there are no clinical for this variant with a microcephaly, there is nothing really important here. And then so we ignored, unfortunately, ClinVar was not very helpful here. So that's why, that's mean we need to go through all the filtration steps. And then we'll start with this case, with the autosomal recessive disorders. Again, we'll be looking for homozygous variants. I will ignore this now, and we'll be looking for homozygous variants that are present because for the rate of high consanguinity in our population. So we ended up with a homozygous variant in autosomal recessive disease that is around 670 variants. And then what is the effect? We want only the loss of function variants, and we have now only 20 variants. And then prediction tools are not very useful in the loss of function variant. And then we did the filtration by the phenotype, a variant that is a variant in a gene that is associated with a microcephaly. From our experience, we used 0.6 as a cutoff limit, and anything that is below than that, the real association, we did some validation. The real association was not really very strong. And if you look into this, so these only the 6 list of variants, or the variant of 12 variants in different genes that's been associated, that one in the gene rank or phenorank is more than 0.6. It's a loss of function variant, and it's a homozygous variant that's been seen in autosomal recessive disease. And we did this and we filtered based on the phenotype. And then if you look into this and you sort by either descending, so what is the most common possible variant or gene that is really associated with the microcephaly. As the number goes up, the association will be higher. For example, here it's like 0.99, and then you go down. If I want to know more about this variant, I just click on here, and then click on this details, and this will load all the details about this variant. It's an ASPM gene. It's a termination, loss of function. If this variant, this is because it's called by a different color. We are missing some of this information. We face it sometimes. But that variant has not been seen any allele frequency. And the OMIM details or the phenotype of this variant is really microcephaly, and this gene is one of the genes that is known to be associated with the microcephaly. And it turned out, well, we know that this variant actually is one of the most common variants in Saudi Arabia that could cause microcephaly. What I did, I just showed you very simple, straightforward filtration and we linked it with the phenotype, and then we sort based on the phenotype association with the type of the variant, and then this was ranked at the top. Again, then what you need to do is just include it in your list, and then you go to your report, and this variant will be included and populated into your report. One of the comments that we are working with Hauwa is based on the ACMG guidelines. Your final report should be either positive if you are able to identify the disease-causing variant, negative if you could not identify any variant that is related to the phenotype. However, sometimes you came across a variant where you are uncertain, a variant of non-significant, and that's where ACMG have one subcategory called, or one category where it's inconclusive results. And that we are working with Hauwa to improve this and include it. Otherwise, it's really very flexible. You can add, remove any of this information. You can create your own template, and some information we really made because we are not doing panel, and panel coverage may be something that we don't want to include. I think that's it. I just don't want to go into deep filtration process. However, I believe like it's a very powerful tool for filtration, but I want to make it how could be a very useful saving time tools from our experience that we could reach the pathogenic variants very easily within a very short time. If you have any question, comment, please feel free, and then I think Hauwa will walk through some report customization.
Absolutely. Thanks so much, Ahmed, for the lovely overview of VarSeq and how you use it in your lab. I'll go ahead and share my screen now. Let me know if you guys can see that. Again, my name is Hauwa. Thanks for joining us today. I hope you guys are all having a good afternoon. Before I get started here, just a quick housekeeping note. If you have any questions, please feel free to enter them at any time in the questions pane in your GoToWebinar window. We'll try to answer as many of them as possible within the hour we have here today, but if we don't get to your question, we will certainly reach out to you via email to answer those. All right, so before I jump into VarSeq, I'll go ahead and give you a brief overview of our company. So we were founded in 1998. Our founder spun off early work at GlaxoSmithKline, and they're currently a key investor in our company. Since '98, we have acquired many customers globally in over 200 organizations, including top-tier research universities like Stanford and Yale, that's here in the States, major government institutions, and pharmaceutical companies, as well as clinical labs. Just a quick overview, we started out with Helix Street, which has now become our SNP and variation suite that's more geared towards the researchers. We also have GenomeBrowse, which Ahmed showed you a little bit, which is our high-power visualization tool. And then, of course, VarSeq, our very powerful filter and an annotation engine. So with over 18 years in the industry, we've become quite the thought leader. We publish a lot of ebooks, and blog posts and industry topics and things like that. So of course, when you buy a Golden Helix product, you get way more than just the software. We offer unlimited training and support for all of our clients, often with a turnaround time of less than 24 hours. So if you have any questions ever when you're playing with the software and anything arises, we're just an email or phone call away. All right, so let's talk about VarSeq. As you probably can allude, VarSeq is our powerful commercial-grade software. It's designed for your local hardware, so it's not a cloud-based software, so you can avoid all the security issues that may arise with cloud-based softwares. It's very flexible, as you can see already. And we start with importing of your VCF files from your secondary pipeline. And then we can add variant and gene annotation sources as well as algorithms like full rank to rank your phenotypes based on genes that they may be closely associated with. Now we do have some more packages that could be added onto the VarSeq platform. So first we have VS Reports, which you saw a little bit here today. It allows you to flag your variants from a filtered set and then include them in a customized clinical report, and we'll go over that here in a little bit. Our reports can be rendered in either HTML, PDF, or XML format. And then next we have our VS pipeline, which allows you to completely automate your workflow. So if you're a high throughput lab and you need to do a lot of high throughput analysis and you don't really want to go into VarSeq, the interface to do a lot of your filtering and annotation, that can be automated with our VS pipeline tool. And then I don't know if you guys have heard, but we have recently released our latest edition, which is the VS Warehouse. That is our data warehousing solution that not only serves as a data store, but also allows you to organize your samples into projects and then use those projects as annotation sources in future VarSeq projects, so you can keep track of variants over time as you get more and more samples coming into your lab. The warehouse also serves as a centralized host for your VarSeq reports that allows you to upload and update reports and report templates for your organization in one centralized location. Being a data store, it was obviously built with scalability in mind, and it has multiple interfaces with which you can connect to it, and it also comes with its own API. All right, so now because VarSeq is a filter and an annotation engine, it's of course only as good as the annotation sources that supports it. And so VarSeq is backed by an extensive list of curated data sources that includes things like dbSNP, Exact, ClinVar, Cosmic, and many, many more. We're very proud to have one of the largest and most up-to-date repositories of public annotation sources. And our data curation team takes a lot of pride in the attention to detail and making sure that the data you're using is accurate. So if we ever find problems with any of the data sources, we certainly will reach out to the data source provider to fix it before we make those annotations publicly available. We also, as you can see from Ahmed's presentation, we have OMIM that's available to add onto your VarSeq license, and then we also have our OncoMD data sources as well as our CAD scores. Now, some of our sources are updated fairly often, like ClinVar and OMIM are updated on a monthly basis. So you can see that information can change from month to month as those data sources get updated. And so we don't automatically update those sources in your VarSeq projects, but we do give you the opportunity to update as those updates become available by giving you a little update button. So we make that easy for you. All right, so to get you up and running as quickly as possible, that's always our goal. We ship with default workflows for things like tree analysis and hereditary gene panels and somatic mutations. And from these default workflows, you can certainly customize those workflows to make it your own and fit, what your lab is doing and things like that. We also ship with example projects in case you want to try out VarSeq, but don't quite have data available to you yet. Now, we do ship with 3 report templates. We ship with a hereditary report template, a cancer report template, and then one for exome trios. And like Ahmed said, our report templates are very customizable. We're always willing to work with you to make those reports look and feel and act the way you want it to. So that's certainly something that you can reach out to me for more information about. Generally, we try to stick to the ACMG guidelines through the VarSeq interface, but you're able to change things like your logo, your lab information, the test descriptions, disclaimers, and things like that. We try to make the generating those clinical reports as easy as possible for you. So as you saw in Ahmed's presentation, it's as easy as using a Microsoft Word document and pushing a button. So if you ever find problems with that, certainly reach out to us. As you'll see during our demo, I'm going to give a brief demo here in a few minutes. The information that goes into each report section can be changed and customized as well. So again, for our high throughput users, our VarSeq pipeline offers a way to run samples through the workflow in an automated fashion. So once you've saved a VarSeq project as a template, and I'll show you how to do that here in a few minutes, you can then use that particular template in a VS pipeline command line environment. And essentially all that does is it runs through your workflow as you would, as if you would on your VarSeq interface. Automatically adds the annotation sources that you set in your template, does the filtering for you automatically, and then you can even export the results to either an Excel file or VCF file, or even start generating a clinical report, which then your clinician can go into and verify the output is correct before signing off on it. All right, and then our VarSeq warehouse interface is highly integrated within the VarSeq platform. Again, it serves as a place to archive your full VCFs of every sequence sample you've ever seen in your lab. You're able to query that particular interface and retrieve subsets of data whenever you want. You can also use the warehouse to ask things like, have I seen this variant before in my previous test samples, by annotating against your warehouse data source. You can tell at what frequency have you seen that. You can ask questions like, does this gene contain other rare variants that I have in my cohort? You can even use the warehouse to ask questions like, what has changed from month to month within ClinVar? So it has a variant that I used in one of my reports that was once considered pathogenic. Has that changed over time? Is that now considered benign? Or has a variant that I flagged before as interesting but was considered benign, has that over time changed to being pathogenic? So those are some questions that you can ask the warehouse and get a nice response back from it. All right, so from within, again, VarSeq and Warehouse are highly integrated. So once you create a project in VarSeq, you can automatically upload those projects into the Warehouse and vice versa. You can annotate against your catalogs and your annotation sources within Warehouse down into VarSeq and vice versa. So for the sample that I'm going to be showing you today, it comes from the Illumina tree site myeloid sequencing panel. We're going to be looking at the 10% dilution for Horizon DX known somatic mutation with the NA12877 sample. We have BAM files and VCF files that are available for this particular sample, and it also has very high coverage. So I'll go ahead and switch over to VarSeq just to show you what my interface looks like there. It looks very similar to what Ahmed had on his screen. Here in the right-hand side, we can see our table view that contains a list of all our variants that we've imported for this particular sample. So we had a little over 120,000 variants. And then to the right here, we can see some more information that comes from our sample VCF files, so things like read depth and zygosity and allele frequency and so on. And then as I scroll to the right here, you can see the different annotation sources that I've added for this particular project. So we have things like Cosmic, VarSeq, ClinVar, or OncoMD, OMIM, and so on. Of course, you can always add more annotation sources to your project by clicking on this Add button here in the top left corner and choosing Annotation. And then you can see we've got a couple of different repositories from which you can choose annotation sources from. So we've got a public annotation repository that contains a list of all our publicly available data. And then we also have our secure bucket that has those CAD, OMIM, and OncoMD annotation sources. If there are any databases that you would like to use as an annotation sources that you don't see within the VarSeq interface, just shoot us an email, let us know where that particular data source can be found, and we'll be more than happy to curate that for you and make that publicly available for everyone as well. Of course, that is if it's a public source. And so, you know, we've added our annotation sources, and then we've also done some filtration on this particular data set. And so, of course, we've done our quality control measures to get for read depths greater than 2,500, again, because this is a very high. This panel has reads greater than 3,500, so we just wanted to filter down those that are of poor quality. And then we also added our alternate allele frequency card to select those that were greater than 1%. Anything in your table view can be used as a filter card. All you'd have to do is to right-click on the column header for that particular column and then choose Create Filter, and then you can select the choices that you want to make within those filter cards. So it's very simple to use. So our filtration steps got us down to three variants of interest here. And again, as we can see, we can quickly use the detail pane to see the information that's contained within these particular variants to see if they match what we're looking for essentially. All right, so once we've flagged our, we've identified the variants of interest that we're looking at, we've flagged them as interesting, we can go ahead and then create our clinical report. I have a report template here open. It's slightly different than the one Ahmed showed you, but just to show you the differences and what the reports can be. We have our patient information, reference, and sample information. If you have gene panels and you bring in your BAM files, the panel coverage and the average read depth is automatically computed for you. A lot of this information can be brought in via a text file, so you don't have to type them in all the time if you don't want to. And then, of course, we have the results section that contains our comments and our interpretation summaries as well as our recommendations. And then again, we just selected the variants of interest that we wanted to put in our report template, and then all the information is pulled in from the OMIM annotation sources. Of course, this just serves as a boilerplate, so if you wanted to add more information in these sections, you certainly can do that. If you want to remove information, you can do that as well. Our report templates are very, very, very customizable. The sky's the limit as far as what it can do. We've had report templates that essentially little mini programs in and of themselves. But if you wanted, different sections for your report template, that can certainly be done. If you wanted more than just two sections, so for example, in here we have genetic variants and incidental variants. If you wanted to add a third or fourth, that certainly can be done as well. So I'll go ahead and render this report template, and here's what this particular template looks like. It looks a little bit different than the one Ahmed showed you, and again, that's just a customization to this particular report template, so it's presenting the information in a slightly different way. We've got our lab information, provider information here at the top. We've got our patient and sample information in a nice tabular format. Our positive results were colored red just to make that stand out a little bit more. And then if we were interested in any kind of particular genes that we had in mind, we can flag those and see if any of the variants we've selected appear in those genes that we're curious about or interested in. And so, yeah, so in here we again have a section, a table view of our genetic variants. Again, these can be customized. Ahmed's report template is going to look a little different than this. It's going to have some information displayed in a slightly different manner, and some of this information will be split out to make it a little bit more readable for him. And then once we go to the bottom, we also have a section for references. We've made that a nice pretty format, so it's really easy to copy and paste if you needed to or something like that. Down here at the bottom, this is very important. Once you first render your report, it's going to say draft report. Let's say you're satisfied with your report template and you're ready to send this off, or you're ready to be done with it, you can go ahead and sign off on your report. So your clinician can sign off on that report and say, this is done, we're ready to go, by just clicking on this verify button down in the report template and then re-rendering the report. And now at the bottom, you can see that this report has been approved by myself and the date in which was approved. Again, these can all be customized. Look and feel can be customized. The kind of information that goes in can be customized. So if you have an idea of what you want your reports to look like, certainly just shoot us an email and we'll be happy to take care of that for you. Of course, our report templates, like I said, can be saved as a PDF format, as that's a little bit more easier to email around than HTML and CSS files. So if you just save that as a PDF, it generates a file which you can send to either other clinicians or labs or anything like that. So once you've completed, these steps for one sample, say you wanted to use the same workflow, because now it's a defined workflow, you want to use it for all the other samples that come in, and you don't want to spend the time adding those annotation sources, creating those filter cards and things like that. All you'd have to do is to save your project as a template by choosing File and choose Save Project as Template. It's going to ask you for a name and some descriptions and some more information particular to this template. And once you've saved that as a template, you can then use that template the next time a sample comes in, and all you'll have to do is to import your VCF files and possibly your BAM and text and patient text file. And then all the annotation sources will be automatically ran, all your filtration steps will be done for you, and then the only things left for you to do is to analyze the variants that come out of your filter steps, and then flag those variants and create a clinical report. Now, when all is said and done, if you had the warehouse added onto your package, you can certainly add the samples that you've just analyzed into your warehouse by just clicking on the Tools, Manage VS Warehouse dialog, and then you can select which project. So for our warehouse, we've got a couple of different warehouse projects that we're working on. So we've got one for exomes, we've got one for cancer, and one for cardio gene panels. So this is, again, a cancer gene panel sample, so we can just add our samples directly to the warehouse and then just upload them, and you can see that this dialog is going to start uploading those samples to our warehouse. So now if we go into a warehouse, let me pull that up real quick here for us. We can see, again, we've got our cancer gene panels project. We've got 2,500 samples already in there, and then we can analyze or query any of these samples at any time that we want, because it's all the VCF file and all the information is stored within our warehouse. So I'll kind of wrap it up there. I know that was a comprehensive overview of what VarSeq looks like and what it can do. If you guys have any questions at this point, please type them in the questions pane, and we'll be more than happy to answer those questions with the last few minutes that we have here. And with that, Brendan, I'll hand that over to you.
Okay, thank you very much, Hauwa and Ahmed, for your presentations there. Again, as Hauwa mentioned, if you do have any questions, feel free to type them into the questions pane there on your GoToMeeting handle. We're going to go ahead and respond to those questions via email. But thank you again to Hauwa and Ahmed and everyone who attended today. We'll go ahead and wrap this up, so have a great rest of the day. Thank you.